you all for making the trek out. I'm really excited to be here. Thanks, Bilal, for the introduction. My name is Eva. I'm a new hire into the BME department here at Georgia Tech and also in Emory. And I'm going to talk to you today about some new work and also some work that I did during my postdoc in using dimensionality reduction techniques and other forms of unsupervised learning to be able to understand and interpret large-scale neural data sets. So a lot of the motivation of what I do in my lab is kind of motivated by the fact that over the past decade and even longer, there's been major advances in optics, recording technologies that have produced and started to generate very massive amounts of neural data sets. And so this is something that has produced this sort of deluge of data in neuroscience. And so part of the goal of my lab, so as Bilal said, the NERDS lab, the Neural Data Science Lab, here is to try to understand and try to interpret the information present in these sorts of large-scale data sets. And so just as a starting point, I'd like to just kind of tell you about some of the different data sets that we work on in my lab. So we think about problems both addressing whole brains, so looking at X-ray microtomography at the scale of whole brains. There we're able to see the anatomy of the brain and try to parse out these large data sets and model brain organization and anatomy. We're also really interested in looking at long-range connectivity data sets coming from two-photon, serial section microtomography. And so with here, we're actually able to see the long-range projections in the brain and then be able to understand organization, so cluster and parcelate the brain. At the millimeter scale, so smaller data sets in terms of their volume, we're also interested in looking at electron microscopy. So in this case, we're able to see very fine scale structural details of brains, including synapses and connectivity. And then also at the millimeter scale, looking at data sets coming from the functional activity of populations of neurons, and so things like calcium activity within these populations of neurons. And so we work on data sets both in anatomy and also within functional data, but the topic of today's talk is actually going to be more along the lines of looking at the activity or the function of populations of neurons and understanding how we can actually pull out low-dimensional structure or start to interpret the activity of many neurons simultaneously. And so to that end, in terms of these new technologies that have really started to scale up our ability to record from many neurons simultaneously, there's a lot of new probes both in measuring electrophysiological properties of large populations of neurons, as well as calcium imaging data sets. So this new neuropixel probe, a new paper came out of Genelia just this past year in 2017, reporting on the neuropixel, which is able to, with two probes, record from roughly 1,000 neurons simultaneously over 1,000 recording sites. And these types of probes can actually survey activity from over 5 different brain areas simultaneously. So these types of technology give us unprecedented ability to start to record from many neurons simultaneously. And the question is, how are we going to actually unravel all this activity and put it into a model of function and of cognition? And so to kind of set the stage and understand where we came from in order to understand where we're going, the first thing that I wanted to talk about was just the kind of standard way that we think about analyzing the activity of neurons. And so kind of one of the simplest analyses that one can do at the single neuron level is to map out the tuning curve or the response profile of neurons. And so in the context of visual stimuli, a standard way to do this is by changing, say, the orientation of a given bar, presenting a bar stimulus at these different orientations to a single neuron, and then looking at the cell's response to those different orientations. And so what this gives us, if we look at a single neuron, map out all of its firing rate or the spikes that come from this neuron, is some sort of curve which tells us what this neuron cares about. So in this case, this neuron cares about bars at a particular orientation. And so we can extend this out, this single neuron analysis, to a population of neurons in a pretty standard way. We can now treat each of these neurons as an independent observer and then map out each of their tuning curves independently. And what this produces is a sort of filter bank of tuning profiles that tell us how an entire population of neurons are responding independently of one another. So as we start to scale up recordings, right, an interesting question that's emerged is how can we start to move away from these sorts of single neuron analyses and start to actually extend these to models that can incorporate correlations and interactions between many neurons simultaneously. And the real question is, what sort of models are good for these large-scale recordings which involve many neurons that are jointly sort of participating in some sort of collective representation of a neural stimulus? And so one way to sort of move forward, and something that's been popularized over the past few years, is this idea of representing the joint activity of many neurons simultaneously at a given time point as some sort of d-dimensional or some higher dimensional feature vector that describes the activity of many of these neurons jointly. So what we can do is we can basically look at the firing rate or the activity of many neurons within the population, and each of these kind of chart out a feature or a single entry within this feature vector. And so now the population activity at each time point within this population is represented by this d-dimensional vector in this sort of high-dimensional space. And so by stacking many of these d-dimensional representations together across time, we can basically form this matrix which represents the joint activity over time of a population of neurons. And so one of the interesting observations that people have found is that if you actually apply different types of dimensionality reduction techniques, which I'll go into details very soon, so this idea of these neurons live in this high dimensional space, but you can actually project them down to a lower dimensional space where the structure of the stimulus or the structure of the sort of the task that these neurons are carrying out has some really nice and ordered structure in this low dimensional space. And so I'll talk a bit about ways that we're actually going to do this dimensionality reduction, ways to interpret what these neural activities look like in this lower dimensional space, and thinking about ways to move forward with this sort of idea of modeling joint activity over time. So just to set the stage, so the idea of dimensionality reduction, which I've kind of alluded to, is this idea that we can take a matrix which consists of these sorts of d-dimensional vectors, right? And there's n of them in this case. So we have d neurons, we have n time points. And now each of these population dynamics vectors is basically a point in this d-dimensional space. And now we have n of those points that are going to chart out some sort of structure in this high-dimensional space. And so one of the most common ways that we go about forming some sort of model for these structures is using linear models. And so the idea behind, say, principal components analysis, which many of us are familiar with, is this idea of basically fitting a linear subspace or structure to this collection of data in this high-dimensional space. And so what principal components analysis does is it finds a linear approximation to our X matrix, so our data matrix. And then the main constraint is that the rank or the dimensionality of this subspace or this model is going to be small. So, I've talked about this idea of characterizing many points in this high-dimensional space as living on a linear subspace. So this is one of the most common ways that we think about forming some sort of model, some low-dimensional model for data. But there's many other ways that the structure of these neural activities or just data can live in this high-dimensional space. And so, we can move away from this idea of linear subspaces to a model, a manifold model. So this now, instead of assuming all the data lives on a linear plane, the data actually might live on sort of some smooth, nonlinear trajectory or space. And so in this case, we can use techniques like isomap, which is a manifold learning technique, to basically find these nonlinear structures within the data. Another model that one might use in this sort of setting is the idea of a clustering model. And so I think many of us have tried different cluster analysis strategies, but might not think about it as a low-dimensional model. But basically the idea is that, in whatever space that our data lives in, it could be clustered and sort of clumped together. And so the idea of methods like k-means and other clustering strategies is to basically find the center of each of these clusters in the data and to figure out which of the data points in our set live in which of the different clusters that we see in the data. So this is another low-dimensional model. And now a generalization or sort of combination of both this linear subspace and a clustering model is one of unions of subspaces. And so this is a sort of newer model that people have started to explore. And the idea is now, instead of having a global model that fits all of our data, instead we actually can model our data as living on a combination of different subspaces. And so this kind of naturally combines the idea of principal components analysis with things like k-means or other clustering algorithms. So what I'll talk about now is some ideas or some examples of how we use dimensionality reduction in different contexts, and in particular in two different animal systems, and both in a visual task and also in a motor task. So the first part that I'll talk about is some work that we've been doing in using dimensionality reduction techniques to do movement decoding or understand the activity of populations of neurons within motor cortex. So the standard setup that we'll consider here is a movement decoding paradigm. So basically we are recording from the motor cortex of a macaque. In this case, we're able to capture about 100 to even 200 neurons at a time. So we're recording neural activity by way of measuring spikes or the firing rates of individual neurons, while the monkey is carrying out a motor task. So they're basically sitting with their hand on a lever. There's a screen in front of them that basically displays a given target for them to reach towards. And so one of 8 different targets will be displayed on the screen, and then the monkey will make a reach towards one of those targets. And so while this subject is moving towards different targets in space, we're recording the neural activity. And if we look at the sort of distribution of the movements that this subject carries out, if we just look at a few different reaches or different reach directions, each of these data points is a single point in time. This is, on the x-axis, is the velocity in x and the velocity in y. And so what we're seeing here are the trajectories or the distribution of movements to 4 different reach directions in this case. And each of them are color-coded according to which direction the subject was moving towards. All right, so we can, carry out these sorts of movement tasks where the monkey is reaching to different reach directions. We can measure the neural activity, which is neurons by time, and then we can then reduce the dimensionality of these recordings. And so I think what an interesting outcome would be is if we have a good low-dimensional representation of this movement pattern, we'll actually see some low-dimensional embedding that has the geometric structure of the actual movement task. And so what people have found is that in many cases when we take motor neurons and reduce their dimensionality, you can actually see the shape of the movement in these low-dimensional projections, which is pretty exciting. The problem is that in many cases, we actually don't find such nice, clean structure that comes out of these dimensionality reduction techniques. And so really the challenge in using these sorts of methods in many cases is that the signal and the noise structure often vary. And so what that means is that, in one case where we have these nice, well isolated and structured movements in these low-dimensional embeddings, in some cases, whether or not the structure is nonlinear or whether you have more noise on a certain day, you can actually see a lot of warping and changes in the sort of geometry by using these sorts of techniques. And so we would consider this to be a bad low-dimensional representation. So while dimensionality reduction techniques are a very powerful way of visualizing and interpreting the joint activity of neurons in this sort of low-dimensional space, oftentimes they need to be taken with some amount of care because it really matters what the signal and the noise structure looks like in order to be able to use these techniques to get out some meaningful interpretations of the activity of interest. And so basically, we set off to develop a new technique that would allow us to suss out what are good low-dimensional representations of the neural activity and how do we actually find projections that look like this rather than look like this. And so our solution was to actually leverage the distribution of the known movements. And so the idea is that we know that when we found a good representation for a given motor task, for which we know the distribution of movements, we know that when we've done dimensional reduction, the shape of those movements in this, or the shape of the neural representations in this low-dimensional space should resemble the shape of the movements. And so the idea behind our approach was to basically use something called distribution alignment. The idea is called, so this distribution alignment approach basically proceeds as follows. So I've already talked about this front end, which is recording neural activity, taking those neural activities and putting them into a spike matrix where we have neurons by time, performing some sort of dimensionality reduction, which then leaves us with some projected neural activity. So once we have this projected neural activity, we'd like to then be able to find a way to basically align the neural activity onto a prior distribution over movements, and then be able to assess how well these two match. And this will actually give us a way of doing model selection and figuring out when we have a good dimensionality reduction technique. So the way that we do that is through something called KL-divergence minimization. And so the KL-divergence is basically a way of measuring the distance between two probability distributions. So this is just a synthetic example to start. But basically, we have a ground truth data set, which is the space, this distribution that we've computed over all these different movement directions. And what I'm showing here is the KL-divergence that we measure as we actually rotate this distribution at different angles. And so what I'm showing here is what the KL-divergence looks like as we rotate. And finally, when we hit this minimizer, or the smallest point of the KL-divergence, we actually have a very high agreement between this rotated activity and the ground truth of interest. And so really our goal then is to align our neural activities, or these projected neural activation patterns, by, with our prior movement distribution by minimizing the KL-divergence between the two. So this gives us a path forward. Okay. And so, you know, what's really exciting about this is I'm missing a... Oh, here it is. Somehow when I went back, it showed up again. Okay, so what this enables us to do is, as I said before, is to do model selection. And so what I'm showing here are examples of the embedded or rotated and aligned projected neural activities for different choices of either pre-processing conditions or different types of dimensionality reduction techniques. And so what we have here is the final match, or the best model that we found. But you see this, this match is actually found for a nonlinear dimensionality reduction technique. And what I'm showing here is as we change the factorization method, we're showing what the underlying neural activities look like. So we find in this case that principal components analysis and factor analysis, which are both linear dimensionality reduction techniques, actually end up warping the neural activities, whereas a nonlinear dimensionality reduction technique in this case actually produces us with a nice distribution that aligns well with our underlying movement distribution of interest. And here we're changing other parameters of our pre-processing algorithm. In each case, we can basically iterate over all these different choices of parameters. We can then align them onto our movement distribution. We can measure the KL-divergence, and in the end, we can choose the factorization that produces the best agreement with our movement data. So this effectively gives us a way of sussing out the right sets of parameters and the right different ways of factorizing the data in order to produce this low-dimensional embedding that matches what we believe, you know, the prior over our movements to be. So what's really interesting about this is that it also gives us a way of now solving the decoding problem. So the decoding problem, basically, the idea behind that is if I measure neural activity, can I actually predict the movement or the velocities of the subject's arm? And so in the end, when we actually do this dimensionality reduction, align these two distributions on one another, we've also in turn solved this decoding problem. So in the end, we're able to take some sort of movement distributions and then finally align them and be able to decode or predict the movements given neural activity. And so what I'm showing here are the results that we get for 3 different non-human primates when we do this alignment procedure. In the first 2 columns here, I'm showing 2 different subjects that are carrying out this reaching task that I described. In the 3rd column here, we have a different subject that's doing an isometric force task, and so their hand is actually fixed, but they're moving their wrist in different directions towards a given target. And so what we show here is the underlying kinematics. So that is the pattern of the movements that we're trying to predict or decode. This is the best case linear decoder that we get. You can't see it exactly, but you can see the colors a little more in this plot. And so, in this case, we're able to get pretty good agreement with the actual movement distributions through this linear decoder, which actually has access to both the training and the test data. So this gives us the best case performance for a linear decoder. And then what I'm showing here is a supervised decoder that has a little less information than Oracle, but it still has access to measurements of both the movements and the neural activities, and then it trains a decoder and then tests on some unseen test set. A Kalman filter, which is also a standardly used decoding algorithm within this field. And so then here over on the sides, I'm showing the results that we get with DAD, or this distribution alignment decoder, for 2 different ways that we can actually train this up. And so we're able to then measure the R-squareds, we're able to measure the mean squared accuracy, and also the accuracy of decoding the reach direction. And what we find in general, especially, here I think it's a really nice demonstration of the algorithm, where we're able to actually match the performance of supervised decoders and this Oracle decoder, which has access to the training and the test data set. And we're able to also get very good target accuracy in these cases. So what this basically demonstrates is, we found a way to use a nearly unsupervised method to be able to solve the decoding problem. And so when I'm saying unsupervised, what I mean is that I've reduce the dimensionality of these neural data sets without any access to labels between the movement and the neural space. Reduce the dimensionality, solve this model selection problem to basically try to align these neural data sets onto a target movement distribution. And in the end, I can solve the decoding problem with very little training data at hand. So this is just, to kind of further instantiate the claims. And so here in this case, I'm looking at a synthetic case where we can actually generate the activity of neurons within this population. And then what we find is that as we increase the number of neurons that we have access to, and use this distribution alignment method, as we increase the number of neurons, we get better and better agreement, and we can actually approach the performance of the Oracle decoder, which is this best case linear decoder that has access to the training and test data set. So what this is meant to demonstrate is just that as we begin to scale up our recording technologies and we can actually record from more and more neurons within the population, we start to approach the performance of this best case decoder. So distribution alignment provides a potential way forward in doing nearly unsupervised decoding that doesn't need access to simultaneous measurements of both the kinematics or the movement and also the neural activity. And it also just demonstrates that unsupervised learning or dimensionality reduction provides a powerful way to visualize and to interpret the activity of populations of neurons. So with this, I'm going to sort of switch gears and talk a bit more about some more recent work that we've been doing to extend these same sorts of ideas, or the idea of dimensionality reduction, but now to the case where we're looking at visual data sets coming from the visual cortex of mice. For some reason, there's some weird glitch here. Okay, well, I'm missing a slide again. Okay, it's not popping up. Anyways, so basically the setup here is slightly different, but we're still looking at the population activity of many neurons in the brain. In this case, we're actually surveying the activity of neurons within the visual cortex using two-photon calcium imaging. These are data sets actually coming from the Allen Institute for Brain Sciences Brain Observatory data set. So in this case, we're also able to survey roughly about 200 neurons within the primary visual cortex of a mouse. And so what we're actually measuring in this case, instead of the firing rate of individual neurons, we're measuring the calcium activity of these neurons, which is a surrogate for their firing or for the action potential generation. And so now each of these neurons that we're surveying has some associated calcium signal with it. And so this is just showing some examples of the calcium activity that's generated from a population of neurons using two-photon calcium imaging. And what I'll say, and was on the previous slide, is all of this is happening while the mouse is actually watching different types of stimuli, whether it be different sorts of gratings or even natural movies or natural scenes. And so many of these responses then are visually evoked by different types of stimuli, and this is the sort of population dynamics that results. And so if we zoom into one of these traces and look at the calcium activity of a single cell within the population, what we actually find is that this calcium signal is actually, or can be noisy. So what we're seeing here is a pretty large amount of noise that's sort of the neural activity is riding on top of. And then the other challenge with using calcium is that these recordings are also kind of slow, so they are typically sampled at relatively slow sampling rates, and so a single event or a single action potential can be sort of masked within the calcium activity. And so the solution is to use different sorts of spike inference methods to be able to take these calcium signals and to pull out the individual spike times from them. And so, the idea behind spike inference is to use the fact that we kind of know a forward model in this to model our observations. And so, roughly speaking, the observations that we get consist of spikes that are emitted from a single neuron, convolved with some sort of calcium signal or transient. And so, those are roughly modeled as a decaying exponential waveform. So you're going to take your spikes, you're going to convolve them with this decaying exponential, and then add noise to them to get your final observations. And so during my PhD, I developed a method for doing spike inference from calcium signals. And this is just some example of the results, where we actually have the ground truth spikes that are also simultaneously recorded with electrophysiology. So in this case, what I'm showing is the actual fluorescence signal, so the calcium signal that we want to deconvolve. Here in green, these are the recovered calcium transients after deconvolution. And then along here in red, we're showing the individual spikes that we've inferred using this algorithm. And so what we find is that there's really nice agreement between the spikes that we infer and the actual spikes that are generated according to the EFIS. And so for all of our subsequent analyses, we'll actually use the outputs of these spike inference methods in order to analyze the calcium activity at scale. Okay, so the first thing that we aim to do, so I've talked before about this idea of using dimensionality reduction as a way to interpret large-scale dynamics. So the first step, and just to kind of show and be able to assess the dimensionality of these neural recordings, is to first look at basically the singular value decomposition, or a way of measuring the dimensionality or the rank of all these different neural activity matrices. And so what I'm showing here are the results where we look at different types of stimuli. So we're looking in this case at static gratings of different orientations. The subject is also watching a collection of different natural scenes that are flashed onto a screen while the neural activity is being recorded, in addition to natural movies, which are 30-second sequences of movies that are replayed. And so across these different stimuli classes, as well as the spontaneous activity, we can look at the singular value spectrum and basically look at how these decay as a function of the number of singular values or singular vectors that we include. So as I said, this gives us a way of seeing how low dimensional these different types of stimuli are in terms of the population dynamics as surveyed from the calcium imaging data. And so what we found consistently across multiple experiments and multiple populations of neurons is that these, so here this purple curve that's dotted is the singular value spectrum for the natural movies. And so what we found is that for natural movies, we indeed see some low dimensional activity within these populations. And so this was at least a starting point for us to, kind of dig into this a little bit further and try to understand what's going on within these populations when neurons are actually exposed to these naturalistic movies. And these other ones still do have some decay in them, but we don't see as much of this low dimensional structure popping out. So we're going to focus on the natural movie class in this case. I'm having some issues with my slides not popping up. Okay. I guess I just have to skip every other one then. Okay, cool. So this is the slide that was skipped. So the first thing that we wanted to do is really understand the functional relationships between neurons within this population. So one way that you could see some sort of low dimensional activity pop out of the data is if there's ensembles of neurons, so clusters of, or groups of neurons that all seem to be doing roughly the same thing in terms of their activity. In this case, you would see, in terms of the collected dynamics, a very low dimensional representation. And so the first thing we wanted to do was to understand the sort of correlation structure or the functional relationships between the neurons. So to do that, we take the calcium activity, we deconvolve it in order to get the firing rates and the spike activities of all these neurons. And then we're just going to look at the inner products or the correlations between all the different neurons within this population. So we had a total of 200 neurons, and out of those 200 neurons, about 71 of them actually exhibited very nice, tight, functional correlations that produce this sort of cluster, this clustering structure. And so what I'm showing here is the pairwise correlations for those 71 neurons. And so each point in this matrix is the correlation between any two pairs of neurons. And what we found here are about, so this is about 6 different clusters, in this case 1, 2, 3, 4, 5, 6, where many of them have very tight correlations with each other, but then of course there are some cross linkages across these different clusters as well. And so this is one way to visualize it, and then also, a student in my lab has been also working on different ways of actually visualizing these correlations using different graph analysis methods. So ways of kind of, looking at both these cluster structures and then kind of digging into each of the individual clusters to really see what's going on within the dynamics there. So this is just, for now, a visualization, but we're trying to dig into these relationships further in the future. So, one question that you could have is that, so if I see these clusters of activations, what is the relationship between which neurons seem to fire together and whether or not they're actually spatially distributed in an interesting way? So in order to understand that, the next step of the analysis was actually just to overlay the identity of these clusters back onto the actual spatial dimensions of where these neurons were located. And so for each of these clusters, we have a common color that they're associated with. And now these across the whole population, these 71 neurons are the ones that we found that had these really nice clustered relationships. And then we can actually look at whether or not there seems to be any spatial arrangement of these clusters. And so I would say, at first pass, we still need to understand a bit more and really quantify the spatial relationships, but it seems as though these clusters are sort of distributed across the field of view. So there isn't a clear connection between the functional relationships of these clusters and their spatial arrangement. This is kind of in agreement with a lot of results that have shown that the orientation tuning properties of neurons within primary visual cortex in mouse are actually not clustered together and are kind of distributed according to this sort of, widely spread and distributed pattern. And so the next step was to actually look at whether or not we could see if this clustering property is not representative of the spatial arrangement of neurons, we would hope that there might be something within the underlying stimulus set that's kind of grouping these different neurons together in some way based upon their tuning properties. And so this is just an initial pass to try to understand the mean or temporal responses of these clusters of neurons across time, so across the full 30-second presentation of the movie. And so what we're seeing here are for all 6 clusters, the sort of average temporal responses across all of the activities within a neuronal cluster. And then we're also displaying here the image that this cluster of neurons seem to respond to most preferentially. And so what we're seeing here is that, the activity of these different clusters of neurons seem to be sort of distributed and almost orthogonal to one another temporally. So in particular, we have, a cluster like this that seems to, be very loosely correlated with the other population responses. And so this just starts to give us some idea of what these neurons are responding to in terms of the temporal dynamics. And we need to look more into what are the actual characteristics of these movie frames, or of what parts of the movie these different neurons are responding to. So the first step then, or after kind of seeing that there was this nice temporal orthogonality between the different responses of these clusters, so the next step was to basically use dimensionality reduction in order to sort of understand if there's some structure within these projections and the neural responses. And so this is an example now of using a nonlinear dimensionality reduction technique called diffusion maps applied to these 71 neurons that were revealed through our cluster analysis. And what I'm showing here is a 3-dimensional plot of these neural activities after being transformed through this diffusion map strategy. And the colors here now represent either the beginning or the end of the movie. And so, we're basically seeing a transition from the beginning of when we start the movie all the way to the end, which results in this yellow. And so what we're showing here is a 3-dimensional projection where we can clearly now see that different parts of the movie are now kind of segregated in this neuronal space, which is also revealed even within these 3-dimensional projections. In particular, right, we're seeing very almost orthogonal sort of dynamics between certain parts of the movie, which I think agrees nicely with what we saw in terms of the temporal responses of the neurons as well. If we dig into this a bit more, of course, we can see that at this segment of the movie where we see this sort of green, or where we're visualizing the distributions in green, we see that across these 6 different clusters, only one of the clusters is really actively responding to that portion of the movie. And so it makes sense then that we see that the embedding or the reduced dimensional projections of these neural activities seem to be sort of orthogonal and not highly correlated with the rest. And once you see in these sort of other areas of the beginning of the movie where there's a little more correlation across the different clusters, you're also seeing a bit more kind of spread within these projections. So this is just one way of starting to, dig into all these neural activities and understand what's going on within the population. So now, if we do the same thing, but we use all 212 neurons, we don't get as nice of a story. What we're finding here is that, this nice kind of green and orthogonal cluster within, that represented some portion of the movie has now been sort of collapsed. We're starting to see that the neural activities when representing the beginning and the end of the movie are sort of in the same position in this lower dimensional space. And so what this suggests is that, if we look at all of the neurons within our representation and do dimensionality reduction, we actually might see a degraded response in terms of our eventual embeddings. And so to kind of dig into this a little bit further, I'm showing, and so the title of this slide is Not All Neurons Are Created Equal, because I think the sort of idea behind a lot of this is that, just because you have more data or just because you have access to a full population of neurons, it doesn't necessarily mean that all of them are working collectively to encode the same piece of information. So there can be benefit in actually subsampling your data set first using this sort of cluster analysis and then zooming in on a subpopulation within the network and then looking at their neural responses. And so what I'm showing here is basically the reduced dimensional representations of the 70, just using those 71 neurons, which were revealed by the cluster analysis on a training set. So this is just the first 4 presentations of this movie. And then this is, if I take the mapping obtained by these first 4 trials and then try to project them and predict on a test set, so the remaining 6 trials or presentations of this movie, this is, the subsequent embedding that we get. And if we try to do the same thing for the whole population on the same 4 for our training and then the remaining 6 for our test set, this is the result. And so what we're finding here is 2 things. One is that, we have a much better way of segregating these different time points in the movie if we use this subset of 71 neurons in comparison to the full population, but also the embeddings that we are getting are actually more stable across the training and the test set. If you compare to the training and the test set across the full population, you see a lot more scatter and you also see a less stable representation. And so what this, just in terms of a hypothesis or what we might take away from this is that if we use a subset of the population, we can both get better segregation of the different directions, and we can also get a more stable embedding and predictions at subsequent trials. So, the last thing that we sought to do in this case was to think about new ways of actually visualizing these mappings or these low dimensional projections of the data. And so what this is showing is basically, so we're taking a 3-dimensional projection of this neural activity into this low dimensional space, and then we have a mapping from the 212-dimensional space into 3D. And so now for each of these neurons, I'm basically color coding them according to the coordinates of this projection or the coordinates of this mapping. And so basically to interpret this, neurons that have similar color are ones that their neural activities are combined in a similar way to produce this sort of lower dimensional embedding. And so what this is showing is that perhaps instead of thinking about, these clusters of neural activations being distributed spatially, instead, maybe it's that the way in which multiple neurons are combined could actually be distributed spatially. And so I think in this case, we're seeing a much tighter agreement of clusters within neurons of the same color here. So roughly speaking, what this says is that this potentially gives us a new way of thinking about the spatial arrangement of neurons within the cortex by using dimensionality reduction to sort of visualize how their neural activities are being utilized to produce some sort of output. And so this is, I think a kind of cool plot. And so we need to dig into this a bit further and really see if indeed these spatial relationships are preserved across many experiments and across different populations within different mice. Okay, so in summary, the overarching sort of idea behind this was the idea that dimensionality reduction can be used to understand large-scale neural activity. I walked through two different examples of how dimensionality reduction can be used to analyze neural data. The first was that in movement decoding, where we showed that movement priors can be used to guide factorizations of the data, and then also to be able to actually decode neural data without access to supervised data sets that contain both the the movement and the neural activity recorded simultaneously. The second example was that of some newer work in being able to use dimensionality reduction for studying visual coding. And so some, sort of summary points of that was that, using kind of combinations of clustering or subset selection along with factorization can be a useful way forward, and that maybe not all neurons are created equal when doing these sorts of analyses. It can be useful to subsample your data first and then do dimensionality reduction. All right, and so this is the NERDS lab, the team that we've just over the past 4 months, we've gotten a lot of new students in the lab, very excited to have them. A lot of folks here have been working on some stuff related to this, also dimensionality reduction for different data sets. And then Aish here was working on some of the new ways of visualizing functional connections within the data sets that I presented in the second half of the talk. And also I want to thank my collaborators. So the movement decoding project included my postdoc supervisor, Conrad Cording. The data sets that were collected from monkey cortex were coming from Lee Miller's lab. And we were also working with Mohammed Azar, who's now at DeepMind, on a lot of the analyses. And then also at the Allen Institute, Saskia de Vries has played a role in kind of guiding some of these analyses of the optophysiology results that I showed. And with that, I'll thank you and open up for questions. Thanks. Yes.
So for the second study, the point we're making about not all neurons being created equal.
Yeah.
To what extent do you think the profile you showed us when you went to all 212 cells is driven by neurons that either have, say, a broad field of view, if you will, across the task, or maybe they have multiple firing fields, if you will, so they respond to two distinct components of the study. And so when you look across the whole, in the attempt of parsing the stimuli, it looks like it's a bad neuron. But it's actually could be quite informative if you zoom out from the individual stimuli.
Yeah, I mean, it's a really interesting question. I think that, so yeah, it depends on what you're trying to do, right? What you're trying to decode, and in this case, for this particular way of looking at it, right, in terms of discriminating across different parts of the time series, they seem to kind of, combine different pieces of information in such a way that it was a little bit less easy to suss out the different time points. So I think that's part of it. They could be tuning to multiple different features in the image, and so in that case, these reduced dimensional projections aren't really conveying what they care about, right? I think the other part of it too is there can tend to be a lot of noise within the data, especially within calcium imaging data. So when you actually look at a lot of these neurons and their responses, it seems as though they're not strongly tuned to many things. So it could be a combination that they have combinations of tunings, and then also that they're just firing, and kind of not responding to the stimulus, and it's kind of more of a spontaneous activity that we're picking up on. So it's kind of hard to really tell from this, but I think looking at a lot of these neurons, it seems as though they're not responding as strongly, and so being able to throw them away allows us to actually zoom in to what we're looking at in the task information in a more clear way. But yeah, I definitely think that there's some probability that they're combining information. Also, we're reducing this down into 3 dimensions. So maybe they are giving some useful information, but actually it's in some higher dimensional space from this 3D plot. So yeah, that's a good question.
I was wondering whether the stimuli, the video stimuli, has any kind of flow itself, like so that subject can detect whether this is the beginning of the video or the ending of the video, like that.
So flow in terms of the time series, or?
Yeah, in time series. Or was it just randomly showing, random?
Oh, yeah, no. Yeah, so the video sequence is, playing continuously. So it is, yeah, it's not just shuffled around, right? Everything is moving within this flow or this stream of the time series. And so, yes, certainly previous time frames should inform, whatever stimulus is going to happen at the next time point, because it's sort of smooth progression through the movie. So in that sense, yeah, there should be some correlation between subsequent time points. I'm not sure if that's...
So the subject can actually detect whether this is kind of ending part of the video?
So, I mean, what the analysis shows is that the representation of different time points seems to be sort of separable, right, in the neural activity at the large time scale, or sorry, across the whole time period. And so, it's likely that what these neurons are responding to is driven strongly by frames or, parts of the movie that happen either at the beginning or the end of the movie. And so, yeah, based upon those properties, you can actually zoom in and see which part of the movie that you're seeing.
So, I know the point of the kind of analysis you're doing is to be kind of agnostic and say, let's just throw an algorithm at it. I just want to hear your thoughts, just maybe more philosophical, about how you think about, for example, in the vision examples you give, there is some retinotopy to it, right? And so there's some constraint there about what neurons will tend to fire in response to versus not. But at the same time, you're trying to be agnostic. So I'm wondering how you think about sort of the balance between this agnostic view of like, let's just let the algorithms figure it out versus imposing some kind of constraints that might be reasonable. Is there a way to do something hybrid between those two?
Yeah, that's a good question. So, I mean, in terms of the retinotopy, all of these neurons are actually at the same position within the retinotopic field of view, right? So it's a 400 micron in X and Y, so 400 micron by 400 micron field of view. So all of these neurons are roughly within the same position, or, attending to the same position in space. But so, in terms of other properties that you could kind of constrain, right? So using things like oriented bars or drifting gratings, you can start to characterize the tuning curves or what these different neurons care about using these sorts of approaches that I discussed at the beginning. So what comes out of that is actually not very clear. So we did look at that. We looked at what are the orientation tuning of these different neurons as a way of trying to figure out, what these neurons seem to care about. And I mean, so in the very simple case where you just have a single oriented bar, a single angle, you can map out the tuning curves. In a lot of cases, these neurons actually care about multiple different orientations, right? It's not very clear to see, a clear distribution in what this neuron cares about. And then you go over to the naturalistic stimuli and kind of things are a lot less constrained, and all of a sudden there's many different orientations within the field of view. And so I think, while the constraint setting can be in some cases very useful, right, to guide these analyses, I think in this case, looking at the orientation tuning wouldn't tell us which of these neurons are naturally clustering together in terms of their actual dynamics. And so, the next step here, or a hybrid approach, right, could be once you figure out which neurons seem to be firing together, can you then go in and kind of look at these properties of those neurons and try to understand how they're distributed in terms of their orientation tuning and what they care about. I think it's not as simple as, neurons care about this one thing, right? They're actually kind of working together to encode some more complex properties of the stimuli. And so what could be is that each of these clusters kind of has the ability to represent many different orientations and many different types of stimuli. And then you need many such clusters kind of distributed around the cortex to actually produce a sort of robust and redundant representation of information. But I think, yeah, a way to kind of combine the approaches would be to maybe take a more agnostic approach and then go and dig in and see if some of these other analyses can tell us something to add, or going, vice versa, right?
And if you had a much bigger field of view, it's coming, right? The technology's not quite there, but if you had like a really big view of cortex with thousands of neurons with the calcium imaging, then probably there's going to be just some physical constraints on parts of the visual field that some neurons are associated with versus others.
Yeah. Yeah, so I think there's been some recent results out of Ann Churchland's lab where they will actually image a larger field of view, or just a field of view that's kind of on a boundary between two different visual areas, say Vis-P and higher visual areas, and you can actually use cluster analyses to really pull out spatially defined clusters of neurons that seem to be kind of working together in Vis-P, which there is a clear, discrete boundary between those neurons and neurons on this other, side. And so, yeah, I think once you start to zoom out, right, it does make sense that you should be able to find representations that seem to be more coherent depending on which brain area you're in. And I think, an exciting thing that I didn't talk about as much in the summary is this idea that I described at the beginning, which is this unions of subspaces, right? Which is kind of combining these low-dimensional representations with clustering ideas. And I think for once you start to zoom out and have many more neurons, maybe instead of finding one subspace or low-dimensional model to represent your data, you actually need more than one. And that could be one way to kind of use these techniques, but to understand the activity of much larger populations of neurons. So, yeah.
Wondering if there's a chicken and egg problem here, where you can use these techniques to extract low-dimensional structure if there's already low-dimensional structure in the data, right? So part one, the movement is really
Low D, yeah.
right? One thing basically, it's a lever. And the plot you showed with the singular values also showed that the one that had the lowest structure was the one that has natural motion, which is like a big constraint in data compared to spontaneous activity, which has no motion, right? So I'm wondering what your thoughts are on how these approaches could give us insights about low-dimensional structure in things that are already high-dimensional, like multi-dimensional movements or a different kind of visual stimulus or something.
Yeah, it's really interesting. So there's been some work that's been done recently, to try to characterize how the dimensionality of the task at hand, right, kind of scales with the dimensionality of these neural representations. So it is true, when you have very low dimensional and very constrained tasks, you're going to see relatively low dimensional neural dynamics, right? And as you start to introduce more complexities, it makes sense to, also see increases in the dimensionality of the neural representations. So, there's a number of different things. One thing is, do you try, perhaps when you look at something, in a complex task and you look at its dimensionality, it actually appears to be high, but it could actually be a combination of many low-dimensional structures. So that's one case that one might, think about trying to start with. And so for that case, this idea of the unions of subspaces, right, or these models that try to decompose some very complex dynamics into a combination of smaller pieces, right, each having low-dimensional structure, could be a path forward. And it is true that if you were to look at them all and try to just do a gross analysis of it, it would seem very complex, but it actually consists of many simpler parts. So that's one thing. I think, as we move towards naturalistic stimuli, we're clearly going to see complexities arise that are kind of hard to wrap our heads around. And so maybe there's some way to kind of move between these two spaces, right? Where you're kind of gradually increasing the complexity of the stimulus in a sort of systematic way and try to understand how that starts to increase the complexity of the neural representations. So I think both having combinations between, naturalistic, where there's no parameters, and having things that are nicely constrained and parameterized is one way, or then also using these kind of mixture models where you're trying to find multiple low-dimensional representations in the data. So both of those, I think, ideas could be a path forward. All right. I think we need to cut it off, but I'll be here afterwards.